interactive. And so feel free to pop something in the chat or anything like that. Happy to oh, wait, okay. Happy to elaborate on something more, and I'll make it a little interactive, kind of. You'll see what I mean. But I'd like to clarify that. So, in addition to being a biology professor at Salisbury University, I'm also the scientist in residence at the Rommel Center for Entrepreneurship, part of the Purdue School of Business at Salisbury University, due to my kind of entrepreneurial endeavors to my artwork. So, through art that I developed in this really odd way, where this is not, if you tell somebody this, how to become an artist, everything I did is not how you do it. But through this weird way, I'm now an entrepreneur now, working to use art for outreach and expanding my own business with that. I realize, well, I'm going to flip back here. My slides are a little out of order. I guess I deleted my intro slide. Anyway, basically, we got the introduction already. I've done various work on amphibious fishes, hagfish slime. Some work on my lab is currently doing is on invasive species, trying to look like the blue catfish and northern snakehead. We're doing some removal efforts and studying their diets, impacts on the ecosystem. And here is like a 47-pound blue catfish we got out of the Nanticoke River. And this one had an adult wood duck in its stomach, showing that they're eating everything and anything and everything out there. And I'm going to come back to this mostly digested duck later on in the talk. You'll see where it connects. And if you want to learn more about my talks, want to learn more about what some of my other areas like the amphibious fish stuff, you can check out some of my TED Ed lessons. Like on, I made one on hagfish slime and one on amphibious fishes. Well, we're gonna jump off into. Ooh, it's a little delay on my end. It's into my artwork. So, in addition to being a scientist, I'm also a scientific artist, and I like to use art to highlight fish anatomy and diversity, including those big and small. So, can anyone guess any of these four fish that I put up here? Feel free to put it in the chat. See if anybody can get them. They're all local species to Maryland. All of them I caught on the eastern shore, but there's no reason that they shouldn't be found on the western shore or in the bay. Let's see if anybody got any guesses. We got some guesses in here. Flounder. So anybody know? Yes, it is a type of flounder, but can anyone guess what species of flounder? It's a little unusual one that people don't even realize we have around here. So it is a smallmouth flounder compared to things like summer flounder, winter flounder, that may be more common. Summer flounder being fluke. Those have much larger mouth. These are smallmouth flounders. They only get to about 6 or so inches is typical. So they're quite small and got small mouths, so people don't usually catch them. Some of these other ones in here, we got a bluefish that's a little bit more recognizable, a larger predatory fish. We have a sheep's head minnow, which are often caught in minnow traps, and you can find them in like the tide pools and salt marshes of the Chesapeake Bay and coastal Maryland. And we have a harvest fish, or a what's it called? A spot butterfish, and this one is actually less than an inch long. And a lot of these fish, bluefish may be recognizable, but using this technique, I can highlight the anatomy of fish that are even less than an inch big, blow them up real big, and blow them up real big and highlight anatomy and diversity by showing people fish in a light that they may have not seen before. And so while flounder are primarily saltwater, the eastern shore does have plenty of salt water. It has the Atlantic Ocean on the Delmarva Peninsula, and the eastern shore has a big portion of the middle and lower Chesapeake Bay, which is pretty salty, and it's got those estuaries too that some of these fish go up in there. But sometimes fish can surprise you. We were beach seining in an area that was pretty much just fresh water in one of the tidal rivers on the Nanticoke River, somewhere between Sharptown and Vienna, if you know where that was. And we happened to get a 12-inch summer flounder in a beach seine in pretty fresh water. So especially these fish that live in estuaries, can have a nice, strong, can have a wide tolerance of salinities to factor in rainfall coming off the coast and things like that. But in order to make the art, it's a complicated process that I use. that I learned this technique from Dr. Adam Summers at Friday Harbor Labs in Washington State. And he is another fish biologist that also wears fish shirts. A lot of us wear these fish shirts, but who's also got a nice passion for fish as well. And the summer after I graduated college, right before I started my PhD, I took this summer graduate level fish biology, functional morphology, biomechanics. It's basically a really intense fish course. And in this technique, he taught me this. In this class, he taught me this technique called clearing and staining, which is also known as diaphanization, that works really well to highlight fish anatomy. So basically, the summary of it is you use different stains to stain certain tissues, like cartilage gets stained blue, bone gets stained purplish red, and you dissolve everything else away. I'll explain it a little more depth later, but it's great for studying anatomy. If you want to study the skeleton, there's everything nice and highlighted, and then you get everything else you don't care about, either transparent or dissolved. But he also showed me it can be an art form while teaching me, given he has these beautiful images. Oh, my cats are going to be interrupting many times, but they have these beautiful images. And he did some local art exhibits around town, and so I knew it was like, well, it can be used in art form. My goal wasn't to learn this for an art form, it was just to learn for science. But we'll get to how it became art in a little for me in a little bit later. So uh, this technique called clearing and staining is a form of histology, often used in biology to basically use stain biological tissues and cell parts and things like that, so you can more easily see things that are, let's say, transparent, or you want to know what is cartilage and what isn't cartilage. You can stain so that it highlights the things of interest. This is mostly used on like small microscope slides to highlight things in very small areas, but this technique is applied to whole fish staining. But it's limited to small specimens. Like I can small specimens up to about maybe 6 inches long really can't be more than like a half an inch thick in any dimension because of diffusion limitations. Diffusion gets less effective the further the distance something has to diffuse. So diffusing through a very thin layer works really well. Diffusing through a brick wall, you can't really diffuse there. And so these smaller specimens are the ones I have to use for the chemicals in order to penetrate without having to get really complicated with these techniques. Although I have kind of pushed the forefront of whole fish staining in ways that basically nobody else had done, so I had to try to figure out myself. So you start with a whole fish that's usually fixed in formaldehyde and then preserved in alcohol, like you may have seen some fish in jars, perhaps at the Natural History Museum. There you go. And then I remove the guts and sometimes the scales to kind of make cut a small hole, pull some stuff out to make the process easier. And if they got big, thick scales, I'll remove those scales so that you can see the underlying anatomy. If they got small scales or the scales are particularly cool, I keep them in for some artistic effects that you'll see. So then I use a stain called Alcyon blue, which I have to use in a mixture of alcohol and really strong acid, and that turns the cartilage blue. I will then use trypsin, which is to digest away proteins. And trypsin comes from is found in the small intestine. It's secreted by the pancreas into the small intestine to help vertebrates digest proteins. And so this is actually it ends up being pork trypsin that's commercially available that I use to digest the proteins away, get rid of the proteins that I'm not of interest, literally digesting that. Just like those digestive enzymes digest the duck, I use that trypsin in a more controlled way for very specific durations to dissolve the muscle tissue and digest the muscle tissue to my likings. Then I will switch it over to a more basic solution with some strong bases like potassium hydroxide, and then I stain it with alizarin red, which is a stain that turns bone or calcified tissue red or purplish red. Then I'll bleach away the pigment with hydrogen peroxide, so I get rid of those dark colors that are on the outside that are mostly on the skin, and I'll end in glycerol. And glycerol is a kind of gloopy chemical with some thicker viscosity, and this helps protect the specimen. Now that I've dissolved a bunch of parts of it away, it's less structurally sound, so by having it in a thicker liquid, it helps cushion it and prevent it from falling apart. And glycerol also has a similar refractive index as collagen, which makes collagen appear transparent. And so that makes the connective tissue look like it isn't even there. I don't have to digest it. Uh, I just have to make it transparent. And so you can see in something like this, you could see some of the skin. You can see the remnants of the skin, the remnants of the collagen, but it's made much more transparent through this process. And I'll show you later what it looks like when it's not in glycerol. It's a little different. And this technique is great for highlighting tissues and structures of interest. Like there are the pectoral spines of a catfish that I'm working on. I'm currently working on a blue catfish study, studying how these fin spines will wear as they get larger and older, and the spine dimensions change. And hopefully, publishing this research by the end of the week. But we use this technique so we can clearly make the fin spines visible, show off those nice sharp barbs and those points. And we can measure the points using this, measure the angles of these points zoomed in, of smaller ones to bigger ones to show how the tips may wear down over time as they burrow in the mud, they elbow each other as they're mating, things like that. And it also helps get rid of everything else that you don't want to study. And so that makes it just really great for studying anatomy and functional morphology in general, and functional morphology being the study of how form relates to function. I'll get more on the research side of things later. Let's start off with how I got into this art form. So after I finished that course with Dr. Adam Summers, I arrived at my Wake Forest University for my PhD, and my house looked like this. It was nice and empty and very cold and barren. And I was like, well, I want to decorate my walls. I'm going to go get some art. So I saw that there was an art gallery nearby, and I went up to them. I saw a nice print. I remember a nice either print or painting of an octopus that was like, I don't know, 16 by 20 inches. And I said, I would like one art, please. And they'll be like, that's $900. And I was like, oof, I don't have that money. I'm a poor grad student. And so I was like, well, I want to decorate my walls. How am I going to do that if traditional art's expensive? And I knew Adam Summers turned his images in art, and I liked those images. And I had some cleared and stained specimens. So let me photograph the ones I had and hang those up. So here's the first one I did. It is a northern sculpin. The scientific name is Isolinus borealis. And I took a photo. And as you can see, it came out pretty cool. But it was different than Adam's. Adam had a nice, great camera and LED light tables and photo editing experience. This is my first time with this. I had only taken my only art experience was a one semester photo class my freshman year of high school because we were required to take an art class. And I had an iPhone and a flood lamp that I turned into a makeshift light table. So it wasn't as clean as Adam's, but I kind of liked the effect that this led to, where it kind of looked like watercolor and brush strokes. And I was like, all right, I dig this. Let me try adjusting I've never used Photoshop before, but let me see if I could toggle with it to make the images look a little cleaner or get a better contrast. And then I used a tool that I wasn't sure what it did, and it did this. And I was like, ooh, it changed the color a little bit. I like that. It's basically shifting on the color wheel. And I'm like, all right, let me do it. I was like, ooh, let me keep shifting it. And I'm like, I'm enjoying these colors. And I shifted again and again. And I'm like, I'm really enjoying these colors. And I'm like, I may not have those clean images as Adam at first, but I like this style where it's these bright, vibrant colors, like kind of like pop art, bringing it less from the scientific realm and more into the artistic expression realm. But I liked all these different colors, so I didn't know. I was like, well, how do I choose? I'm like, I don't know. Let me put them all together and make a piece like this. And I called it Isolinus Warholis, like scientific name being Isolinus borealis, after this one after Andy Warhol, kind of inspired, where he's known for doing those multi-panel images of the same thing in different colors. And I'm like, well, I wondered, is this how he decided on those images? He just couldn't make up his mind on which color of the Campbell soup can he liked best or whatever, and he put them all together. And so I enjoyed this, and I was like, all right, now I got to go print this art out. I don't know how I do it, but there is a FedEx office near me, so let me go to FedEx office and print out the nearby FedEx and print out that piece on canvas. And this was in North Carolina in the summer when it was like up 98 degrees and 100% humidity. And this I printed it out around lunchtime. So I was like, all right, I wanted to print it out on canvas to get it nice to look nice on the wall. But I'm hungry, and somebody recommended this restaurant like across the street, Vin 205, part of Wine Merchants. They had a wine shop and a restaurant there, and I was like, all right, I'll go get some lunch there. And while I was there getting lunch, I didn't want my new art to warp in the car because it's so hot and humid. So like, let me bring it inside. And I saw they were taking down the artwork in there. And I was the only person in there just sitting at the bar eating lunch. And I made a joke like, hey, you need some more artwork? I got something for you. And the owner was like, actually, like, oh, yeah, actually, like, we're getting ready to change out the new art, but we don't have any more art. Like, what do you got there? And I was like, oh, here's some of the artwork I just made and printed out to decorate my walls. And he was like, oh, I love that. We'd love to have that up there. And I was like, really? I just printed that as decorate my wall, and it's like, oh, that's awesome. Do you have more you could decorate? We love to feature you like all over here. And here you can see me with my first piece of art that I ever printed out that was meant to be on my wall in grad school with my fiance in front of this at an event we went a dinner we went there, and you can see the art piece there. And I ended up printing. This was the first one I printed. I ended up having to go. I left that one. I basically had to go right back to FedEx and be like, actually, I'll print one more of those out. And had a few other pieces up there. And I later realized that the owner, Caleb, right here of the one of the owners, Caleb of that restaurant, was a avid fisherman, and that's why he liked the fish art. So now I keep going fishing with him every summer. We meet up on the Outer Banks and go fishing. Here's a big stingray, but we get other stuff. This just happened to be the only photo I had of both of us in there. And then I had other artwork around his shop. Ended up having smaller like paper prints that people could buy up the front. And he had the head chef ended up being my best customer. Where half the pieces I put on the walls in there, he'd just buy off the wall, and so then I had to replace them, and then he'd buy some more of them. That worked out really well for me. Um, but that image you see back there also ended up being the cover photo you can see of the this issue of this scientific journal in zoology of research I did on amphibious fishes, on amphibious sculpins specifically, understanding how their behavior and anatomy and functional morphology can assist with their amphibious behaviors. And so in this example, I was able to use my art to help publicize the science. And from that one restaurant, I spread to breweries, to independent theaters, and cinemas, so local businesses, farmers markets, public events, and some galleries. It's really as far as wide as I could reach, as many different people as possible. Eventually, in some galleries and shows across the country, one thing in Canada, some online shows, and I even recently won a international art competition for this image of my harvest fish collected from the Chesapeake Bay in the 2025 Nikon Small World micrography competition. Basically, photographs that you have to use a microscope. They don't have to be a very big, powerful microscope. And this one was basically the magnification was 0.63. So it was, if anything, I zoomed out slightly on my microscope, but it still counted because it was taken on the microscope. But this specimen was still like the size of a quarter. And this won one of the images of distinction. But personally, I really like making my art accessible. So instead of focusing on high-end galleries, I focus on places average people would likely interact with them, like breweries, that get people thinking about what is that? And then when they ask, what is that like some kind of x-ray or something? Like, no, I get to then pull them aside for a quick science lesson, teach them about the technique, and then teach them about the fish they're seeing and their different anatomical structures and how that relates to those lives and experiences of those that fish. And I hopefully leaving them more science curious than they came in. But my art kept evolving as it started off from just a way to decorate my walls and became more than that. So my technology and techniques improved. So here is a raw image. It may not look that great uh, before I do some editing to adjust the contrast, but you can see the details are quite there in high resolution now that I have a nicer camera and a light table. But I'm still getting the exposure worked. But after I adjust the brightness and contrast and stuff like that in the in Photoshop, you can really see those higher details. But It's tricky getting that balance because you are taking a photo of a very small, thin thing and blasting a lot of light through it. And if you put too much light through it, you drown out the specimen. So you really have to edit it a lot in post to highlight it. But I say this photo doesn't really do it justice. In person, they are more vibrant and colorful like that, but it's hard to photograph them without editing them to look like what they are because of the how small and thin they are and how much light passes through them. And so the typical things, like I do the tip, these are the typical things, adjusting brightness, contrast that say like a wedding photographer would do for your photos, which is why they cost a lot more than you would think because they're not just taking photos, they're editing every single image to look their best, just like this. This is likely how Adam Summers got those clean images he used because he had access to good equipment and knew what he was doing in Photoshop. And so once I learned, uh, I got a little bit better with it as well. But I want to take it a little bit further from being the scientific process and more into being the artistic process. I want to make this kind of my own field rather than just another person doing this histology technique and printing out plain photos of it. And I really like vibrant colors. So sometimes I'll go from here and adjust the colors around and make them bright. I really like bright neon vibrant colors, and that's reflected in my artwork. And sometimes switching to a darker background can help me make those colors even pop more. So I do a lot of kind of these inversions to where that neon colors really contrast well on those dark black backgrounds. But I also like to play around with the arrangements and compositions now to compare and contrast fish, kind of further establishing my own style. So these are the two fish in four different color patterns, but highlighting two really different fishes here. And there are two local ones to Maryland. Actually, I collected these ones on the western shore from Nanjemoy Creek, if anybody knows where that is, kind of east of DC and south of Annapolis a little bit. Does anyone want to take a guess at either of these fishes in the chat? One's a little more uh, iconic than the other, but still not the most known fishes. Let's see what we got in the chat. Uh, Common carp? No, but good guess from that large fin spine up there. Without scale, yeah. So these are all baby. So these are all like 1 or 2 inches long, and so because that technique only works on such smaller fish, if I want a bigger fish, I have to use a baby one. But if I want a small fish like this one that doesn't get bigger, then it works great. So here is an adult inland silverside. This one, a local small little bait fish that you can get in in many creeks and rivers and even some brackish portion of tidal rivers. Schooling fish that a lot of things eat. And these are white perch. So this is like a 1 inch long white perch, but they get up to, I don't know, like 18 inches, something like that. People, I'm sure somebody here has gone fishing for them. I go fishing for them sometimes. They're tasty fish, close relative of striped bass. You can kind of see the resemblance there. And so I like to use these images, comparing and contrasting without kind of indicating a scale bar or anything for scale because it gets people thinking in different ways and sees these fish in different light. By blowing up a fish that's normally 1 inch long into a 36-inch long canvas, you're gonna you're gonna pay more attention to it than if it's 1 inch long in a creek. But also, I can shrink down a big fish into a small fish and give it the same amount of attention. So it's fun playing around with the composition. But this piece was the inspiration for this came when I was doing my invasive amphibious doing my research on the amphibious behaviors of invasive snakeheads back during my PhD in 2017 in the Cedarville State Forest in conjunction with the Maryland Department of Natural Resources. You can see what we were doing, some of that research right here, where we're kind of back when I didn't have the gray hairs in my beard. And you can see a little snakehead on the ground, filming how it moves around and describing its amphibious behaviors. But while doing all this work to highlight the invasive species, the ones I wanted to highlight the ones we were fighting for. So I named this piece Jumwai Natives after two of the native fish we encountered in collecting the snakeheads in the river to then highlight what we're fighting for. And I picked these two because they had nice contrasting body shapes that lent well for this interesting comparison I've discussed. I also really like this one a lot, and being one of my, particularly this blue one, right, the blue variation of the white perch right here. This piece in general is one of my most popular pieces, but I really like it because it really resonates with me. And I like it so much I got it tattooed on my arm. You can kind of see it through the camera here, maybe a little bit. And it was my first tattoo because I knew I was like I was always unsure about tattoos, but there's nothing I really ever wanted to commit to for my whole life yet until I saw this and I'm like, I'm never not going to be proud of this artwork. And also trying to explain this technique to somebody who says when I tell them I'm an artist, they're like, oh, what kind of art do you do? I'm like, well, it's biological and involves some photos and some Photoshop, but also a whole fish. And they're like, what? And so now I just have a something I can show to people and explain them in person and give them a science lesson. It actually comes in handy in lecture when I need a diagram of a fish, a skeleton, which happens a lot more than you think given I teach fish biology. Um, but I actually have two examples on my arms now, and one is let me get back there, of this little skate, a fish you can find it keeps going back, in deeper water off the coast of Maryland. They come shallower in winter. We have some other species of skates you can get on the beaches, like clear nose skate. But I actually got a couple of small ones in the lab for research to study their burial behaviors because nobody's ever actually described how they bury in sand, but that's a big part of the life, and we need to understand how they do that to understand more about their natural history. And so with this one, this is also a meaningful one for me because this one was uh, I mean, in addition to being a really cool, just I think it's a beautiful specimen that I got a local artist in Salisbury to tattoo on me with. Again, I really like those neon colors, and so we experimented with some fluorescent inks that make me neon as well. These glow under black light. I got that one on my bicep, but it'll be more difficult to show right now. And so I chose this one because it was really, really clean. It was really clean, um, and it was really clean and this is kind of that raw image where less Photoshop than the others to get it this crisp. And I wasn't able to do this process during the pandemic because it required steady lab access where I had just finished my PhD in April 2020, and then I bounced around to some different labs around the country for a couple of years before I landed in Salisbury University and got my lab set up. And this technique sometimes requires months at a time with specialized chemicals and equipment that I need to check daily, and that was not possible during the pandemic. So this is one of the first pieces that I was able to do again. Uh, uh, ooh, let's see, where did it go? There we go. Some of these are displaying out of order, or no, there we go. They're glitching. Um, Probably because I'm screen sharing. Anyway, this one was one of the first ones I was able to do with my new lab and a new camera, new Photoshop software, new light tables, real light tables instead of my makeshift ones. And look how crisp you can get. I was able to get that. Like this is zoomed in right here, and everything is still crystal clear and sharp here. And I was really proud of being able to get back to doing the art that I enjoy. In addition to uh, in addition to playing around, improving my techniques, my arrangements and composition changed uh, suddenly when accidentally when I was asked to donate a custom piece for a charity auction for an art-based school in Winston-Salem, North Carolina, during my PhD, maybe about a year or two into being an artist, and they wanted a piece from me to both use educationally and but also this would help raise money to support the school. And for this charity art auction, they often pair two artists, unlikely artists, together to create a piece together that they donate. And they paired me up with a local poet named LB the Poet. I was a little confused at first. I'm like, how am I going to work with a poet? I like, does he know about this research technique? I was really not sure what to make of it and how we can collaborate this. I'm like, I just put a cool image of a fish and that's it. But working with him, like he came to my lab to see my process. I came to his poetry slam event to see his process, and helped me. This helped me expand my arrangements beyond just adding what fish I thought looked cool together. We worked together, build a story that told a narrative, um, and the narrative through this piece I wanted to kind of try to tell was kind of highlight on community. Given this is a community event, I want to highlight on the local fish community, local and just local animal community. We've got some frogs in there too, some fowlers, toads, and this also happens to be similar species that are found in Maryland. So we have a largemouth bass here, a gizzard shad. We have bluegill sunfish, red breast sunfish, Fowler's toad, white perch, the blue catfish here. I want to kind of highlight on community and some aspects of like during this time. I think in our town there was some incidences of like a bunch of people were seeing a crime happen and people fighting, and nobody did anything to do anything to stop it. They just kind of watched. And so we call this one predator and prey, as in like the predator, the largemouth bass, is duking out with the prey, the gizzard shad, but they're more on equal footing in here. But they're fighting it out around the remnants of other dead fish in here. And in this, people are the community is just looking about, just you have onlookers, people coming and going, and not really doing anything about it. And so that was the first shot at putting the narrative through that story. And up until this time, I think the most expensive piece I ever sold was a giant 24 by 36 canvas of the Nanjamoy natives piece I showed you a little while ago. And I tried to sell that to the biology department that's chair who asked me to do a piece for. I'm like, hey, how about $100? And she refused to give me $100, instead giving me $200. And after multiple people paying me way more than I was asking for my art, I started realizing that the value of good art and of my artwork, and I could sell it for what it's worth. I don't have to upcharge it extremely like a $900 little piece like the that art gallery was trying to get me to pay when I was a grad student. But I still wanted to make art that was accessible, but enough to make my ends meet, especially as a poor graduate student. This helped. A little extra cash went a long way. And until I only a $200 piece, because I, even though I only asked for 100, was the most expensive I sold. But then in this auction, a live auction was fun. Getting to see my artwork go up 100, 200, 300, 400, 5,000, 500, 1,000, 1,500, $2,000 was quite exciting. That I was able to, people cared about this much about it, given it was, they'll probably pay more because it's a charity event. Paid $2,000 to help raise money for a local school. But it's really empowering for my artistic career, showing people must really value my art if they're willing to pay that much for it, especially when one's selling this high. And there were other things being sold that this was, I think, one of the highest selling pieces of the night, including human-sized metal sculptures and works by established artists. And I don't nearly charge this much outside of charity auction because I still want the art to be accessible. Given I get into making art accessible, so I still make it affordable, but I got to pay my bills still. And after working on this piece, I want to start shifting my direction a bit to make art that brings people together, helps educate people, makes people think, and continue with that narrative through the art. And so I started getting involved with more explicit events that link the science and the art to highlight issues in the community. So there was an example of this Lost Waterways event of Winston-Salem, North Carolina, where they had like a film series and they did a public art project, kind of painting in the streets of the city, wherever culverts ran, wherever small creeks and rivers that were underground and had to be paid over to make the city, showing people all these hidden waterways, these lost waterways of the city that are still connected underground and still play a vital role connecting our community, connecting the ecosystem. And so there I brought some artwork examples to show people about the diversity in these local little creeks using fish that I collected in those creeks. And while they had some of the artwork here, there's a lot of specimens in jars to kind of more explicitly kind of show people, here's what these little fish are, and showing them technique. But it was, people were amazed to see like, oh, I thought there was just bass and catfish in that river. And I'm like, no, there's over 50 different species in the county. And here's just a dozen of them that I'm showing you, like the bluehead chub or the brassy jump rock or the creep chubsucker or the spot tail shiner or the red lip shiner. I'm sure many of you have not even have not heard about a lot of those, but they're all native species that are in the area that people don't, that are underappreciated because they're not a big fish that people go fishing for, but they're nevertheless part of our local biodiversity, enhance the ecosystems and things like that. And so this is one example of these art events that I did, and I use it to highlight the sustainability issues as well and other events. And I then this led to me working with a local person that managed a park to develop a custom piece for a gazebo that was to be built after this event on a park near a school. And I wanted to highlight the local diversity of the fish in that creek, using kind of taking this to the next level. And so I created pieces like this. I created 4 pieces like this. This is a bluehead chub school where each fish was an equal color shift on the wheel from the end one. Each color shift on the wheel going forward. So this one shifted an equal amount forward on the wheel to get to this one, which then shifted to this one and this one. Each one was the same shift turn on that color wheel until I ended exactly the same beginning I started. So this required a lot of math and calculations to make sure I did it right and not just haphazardly. And so this is a bluehead chub school with a couple different specimens I've thrown in there, where it's kind of flowing into each other, kind of that rainbow harmony. Everybody's kind of trying to build that sense of community. And because it was mathematically the mathematical color wheel formula, I was able to have that flow into the next one. And here's a white perch, and that flowed perfectly color wise into the next school, which flowed perfectly into the next school of rosy side dace. And that flowed perfectly in the last school of the bluegill sunfish. And all of those were supposed to be arranged in a circle in a gazebo. And unfortunately, I got commissioned to make this series for a specific park in mind. And I learned now to, if I get commissioned to make anything, ask for some payment up front, because I got ghosted. And after putting all this work, this is probably the most time consuming pieces I've made. They ended up stop responding to me, and I had the artwork, but never got to print it out, share it with people. So I love sharing this piece now because it was a very exciting piece for me to work with. Unfortunately, my parents ended up. I was able to give the piece printed out to my parents for an anniversary gift. But given the nature of this, and it was nearby a school, I call this piece school. Given it's schools of these fish in the local community, all working together right next to it in a creek next to a school. But just like I can use art to highlight science, and science is just and science can be art. Art can also be science. And so I'm going to give you an example using plecos. These are also known as suckerfish, suckermouth catfish. They're the ones that you often see at like pet stores and fish tanks, munching on algae on the surface, and often dark colors, not moving around much. And this one was interesting because at this time, as I was having fun exploring diversity of fishes by staining a bunch of random fish, whatever I can get my hands on, uh, I was just appreciating the unique morphology of all these different fishes and getting to experience them. But then I got to this one, which was a pet of mine that died of natural causes, and I'm like, well, let me immortalize you in art. And I noticed something odd. After this whole process I went through, these lips were still vibrant, bright white, and I had no idea what was going on. So I contacted Adam Summers, the guy who taught me this technique, and he also thought it was weird. And we thought perhaps it could be lipids, some fat tissue in an unusual distribution. And so he suggested another chemical, xylene, to clear that tissue out. So I gave it a try to see if that worked. And here's what it did: absolutely nothing. Xylene did not work. It was not fat tissue. What else could it be? Adam, after talking with Adam, we thought maybe it was elastin, which using a different series of stains, stained black. So look carefully to see what is stained black, to see, are those lips going to be stained black or not? It's going to be very subtle, but no, everything but those lips stained black. So because elastin can be found in fish scales and things like that, but it is everywhere but those lips. So like, what is in those lips? We thought elastin made sense because that's kind of an elastic sort of connective tissue. It's tough but more flexible. It could help make a suction cup. But with another stain, we determined that everything in dark red was collagen, and so those lips were collagen. It's a connective tissue like elastin, but tougher. It's still a soft tissue, but it's tougher. And there was so much of it that even though glycerol normally makes it transparent, there was too much of it to make transparent, and so it still persisted. And I had a different pleco though that was very different body shaped. That also was a pet that died. This one was a rubber lips pleco. This next one I'm going to show you is a loricaria twig catfish, and I also had, and I'm like, did it also have thick collagenous lips like the other one? No. And so you can see this is out of glycerol. This is not in the glycerol. So you still see some of that the whitish collagen around in different parts. But looking at the lips, there's very little in those lips. So what is going on here? Not all these fish have it, only some of them. And I talked to some collaborators that were like, oh, we study the diversity of plecos. So I'm like, can you send me a bunch of plecos and let me stand them around? And so continue with the scientific method. We established observations, unusual issue. We kept asking questions, identified what it was, but then asked questions about why. So we know that collagen, and not all of them had that much collagen, and they're in a big range of collagen, but why? So you could see some had very little, and some had kind of narrower mouths, and some had increasingly strong concentrations of collagen that we ranked from 0 to 5 was how we qualitatively scored it, where you could see pretty much the whole lips and the whole body is collagen in that one. That's the Astroblepis. It is pretty much a waterfall climbing pleco. And so we're wondering, could these differences in collagen and lip shapes be adaptations for different environmental conditions? And it turned out thick collagenous lips evolved multiple times to help survive, to help live in rocky, high flow environments in plecos. So what you're seeing here is a phylogeny that worked with some collaborators that specialize in phylogenies and kind of working out family trees. They take the DNA, they go out in the field, collect all these fish in exotic areas in South America, take DNA samples so they know exactly which ones are more closely related to others, and they construct a family tree. But then we map some traits out on these family trees. So you can see here, red being pretty much no collagen present, to dark blue being a lot of collagen present. And you see there's an uneven distribution throughout here, but this happens to match up with flow environment. So red over here is a low flow environment, and blue is a high flow environment, particularly dark blue. And so Astroblepis, that waterfall climbing one right here, it's got thick collagen, a lot of collagen, and it's also dark blue. It lives in waterfalls and rapids and high flow environments. Those Catastoma, that genus of catfish that I showed you, those rubber lips plecos, those also live in very high flow environments. And so we use this phylogeny to map these traits out because we need to make sure that first, these fish aren't these fish aren't just all having similar. The ones that are closely related have, well, basically, this isn't just a case of one ancestral fish developing thick lips and then all the related fish that descended from it also having thick lips, because that wouldn't tell you much about adaptation. That could just be randomly one got this trait and then it spread it to its offspring. And so by comparing it to both the family tree and to environmental conditions, we're able to see that, sure, other Catastoma that are closely related do have thick collagenous lips, but they also live in high flow environments. But Astroblepis also lives in a high flow environment and developed thicker, more collagenous lips. We have some other species down here that are somewhat intermediate in those collagen and then live in intermediate flow ranges. We had some like Corydoras and these hypoptopapuma. That's a fun one to say, that lives in very low flow environments, and they have very low collagen in those lips. So showing thick lips evolved in multiple times as an adaptation to live in high flow environments, because those 6 suction cups with this tough collagen reinforcing them make those suction cups a lot less likely to fail under these high flow environments, so they can attach better. Whereas fish in lower flow environments, unlike muddy bottoms, don't need as thick, tough lips to hold on, because there's nothing trying to blow them away in the current. And so those other ones didn't need those lips. And so we ended up publishing this work in the Journal of Morphology, kind of showing that the collagen content of these plecos correlated with flow and the substrate, whether like it was a rocky substrate, a muddy bottom, woody bottom, things like that. And its interesting thing is that in this case, instead of using art to highlight my science, in this case, I use my art to make a scientific discovery, which I was able to investigate through the scientific method and publish. And not only does art inspire biology, and biology can inspire technology. So this is a paper that cited that project, optimal design and experiment of bionic vacuum suckers with high attachment properties. And so this is a field called bioinspiration, biomimetics, learning from nature to engineer better and make new technology. So people, based off of my artwork that led to science, are now using this as information to help them develop more effective suction cups that can be used in a range of applications. So that's just an example of how it can be applied. And because art can inform science, just as I showed a few weeks ago, I was actually got the honor to be the first ever artist in residence at the Society for Integrative and Comparative Biology's annual conference to show how it can be done to thousands of scientists in attendance. And I also got to share some new swag. I got to print out some new artwork and test out new products and share a lot of these things that are now available on my website that I posted earlier in the chat, but I can post later for people who showed up later and may not be able to see it in the chat. And all of this is available for sale on my website, either directly or if you see a color that isn't yet on the website or a piece that is on my regular website or social media, but not on my Fine Art America site, you could send me an email and I could put that up there for you to buy. But if a pro tip is if you want to get a big canvas or a big paper or anything like that, just send me an email and I can probably give you a better price directly from me than from that website that's set up automatically. But I'll end on some local species that you may recognize, where and some that you may not recognize. So we got a lined seahorse collected from near Ocean City. This is the sucker disc of a remora, a shark sucker that stick to sharks and rays and things like that. And what it is actually it's a modified first dorsal fin. So you may see like on a striped bass, like over here, you got the spiny dorsal fin over here. You can see the spines running along down the middle of this dorsal fin. This is a top-down view, but those spines have adapted to basically grow wide, and it may be hard to see, but they have these kind of tooth-like combs around here to help them grip on, and they have a little lip of collagen that you could see, like a little halo of collagen around there that help make a suction cup to help them better stick to sharks and larger animals to hitch a free ride. We also have a cutlass fish, or also known as a large scale hairtail. People often call these ribbon fish in the bay, but ribbon fish are, it's actually a little inaccurate. Ribbon fish are a totally different group of fish that look similar and long, but they're in no way related to these groups. We got a northern sea robin over here. We have, again, this is that cutlass fish, the tail of that seahorse, and a summer flounder here. Again, note that really big mouth compared to that smallmouth flounder, and that's because they're a predator that gets larger and eats fish. And we got some more exotics. And anytime I've had a pet fish or even a frog that died, this is an African dwarf clawed frog and a silver dollar fish. I stained them and added them to the teaching collection I use to teach anatomy and diversity. And so there's some of those examples. With that, I'm happy to answer any questions people got.
Wow, thank you so much, Noah. If you want to come up, yep, there we go. Let me put a spotlight on you.
I'll repost those links in the chat for people who may have come later. If they want to look at more of my artwork on the website or anything. It's also on my social media.
And as always, if you have a question, you can raise your hand or put it in the chat box. It looks like Sarah is putting, I'm curious to hear how your staining and photographing techniques could possibly be used for natural history documentation, similar to how museums have wet specimens, taxidermy, fossils in their collections. I think you touched on it a bit.
So I will say that so these are natural history specimens. These are part of these are part of collections. After I stain them, they go back in the collection to use as references. And so they are a wet specimen. It's just the liquid's different. They're still part of these collections, and they can use to serve a record of what species were around and the morphology. But it's just basically putting a filter on these specimens to be able to use them in a little different way. You lose some data out of them. If you take the guts out, you can't find out what's in their, you can't learn about their guts anymore. So I only do this either with specimens that I collect or specimens or specimens that I collect in what's it called? Or that I need for research. So those plecos, some of those were really rare specimens, but because we had a good research go on mind, those collaborators were able to send me specimens that were cataloged in like the Alabama Museum of Natural History, and I stained them. And then after this research, they weren't to be used for, they weren't my art artistic products. They were still natural history specimens that I sent back to the museum and are still available in the museum today. Um,
And to follow up on that, Sarah, we would love at NHSN, we would love to have a display of cleared and stained uh, uh, items in our collection, or to display, for the public to see, because it's such an amazing way to interact with and learn from the animals.
I could probably help you out with that if you message me later. We could figure that out. Maybe if you can, well, we could figure that out. But certainly, especially if y'all have some specimens to provide. There's rules about if I collect something with a scientific collecting permit. There's rules about how I can share it around. But if y'all collect anything, or if anything, your collection or things like that, I may be able to work around it, or collect some under certain licenses I can use to make that work.
Yeah,
Yeah.
we have licenses. So Ned wants to know: have you done a needlefish or a swamp pickerel? Um,
Yeah, so I've gotten a needlefish, Atlantic needlefish, when I was collecting with the Maryland Department of Natural Resources 2 summers ago. We got a beach staining in the coastal bays by Ocean City. Um, that specimen was like a foot long and it's a little big to stain. So I just had that as a teaching specimen to teach about local diversity. But I'd love to get a nice tiny little one that I could stain in there. And I'm I'm not sure what you mean by swamp pickerel. There's a few different terms that there. I think you probably referring to common names. Sometimes are tricky, or there are multiple common names for the same thing. But I think you may mean redfin pickerel, or grass pickerel, one of those pickerels. But I have collected both of those with somebody that I collected both of those. Unfortunately, the ones I collected are too large to stain, and the ones that I got are too smaller to stain are on loan from another museum, so I can't, like, I use them for educational, so I can't stain those. But I really want to get a small pickerel because I think they'd be really cool to do that. Interestingly, I collected those larger redfin pickerels um, while helping a visiting scientist on the Delmarva Peninsula capture pirate perch, another unusual fish that we are able to, in collecting these fish, we were able to describe a new species of pirate perch that's found in this region, and got some specimens out of the collection after that.
cool. And it says, do you use any other programs besides Photoshop?
Um, Not really. No. Maybe if I'm like posting something to Instagram, maybe I'll use a little filter. But I no longer really do that because I can get everything what I want them to be in Photoshop. That I don't need to rely on filters because I'm already editing them the way I want to edit them.
Yeah, and Alan's asking about. I think it was the skate picture.
Mhm.
Um, when you zoomed in, there were like these star-like pink features on that, and it's on your tattoo too.
Mhm.
So what I wanted to know what they were, and then he
Yeah.
wants to know, is it something that you added with Photoshop as an added effect?
So 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 no, I rarely add anything that will alter the biology. I never add anything that will alter the biology of the fish. And some of those ones, like in Predator and Prey, you saw like the ring around the fish. That was the watch glass, the dish that I photographed the fish in. And so I kind of I made like a little shimmer effect to kind of distort it and kind of make it look like a more violent arena for the predator prey interaction. But I never alter the composition of the fish. I want to highlight the specimen. The nature is beautiful itself. I want to highlight the beauty within the nature. Those little pink stars are the dermal denticles, the scales of skates, and so they are mineralized. They're sharp. They're essentially structurally very similar to teeth. So just like sharks are coated in like their scales or like teeth, these more interspersed scales are sharper and more teeth like to use as defense. And those stars kind of are like kind of a flaring out base of where they attach in the soft tissue. And normally you think of skates and rays and other cartilaginous fish as not having bones. They have they have a skeleton made of cartilage, and they don't have bones, but those tissues still stain red because they are um, and some of their other tissues stained red because they are calcified cartilage. Oh, other tissues are calcified cartilage that's stained red, like in their pectoral girdle that helps with their pectoral fins. But because the structure of teeth are calcified mineralized tissues, they also and those dermal denticles are like teeth. Those stars also stain red like bone, and that's what those are. I was just able to get really zoomed in and get really good detail on there.
Well, I was just to follow up on that. Have you done any baby sharks or
No, I don't have any baby sharks in my collection. I haven't collected any, but if anybody wants to give me some baby sharks, I'd love to stain some and take some photos of those. Um, the problem is you often have to get, uh, given how big baby sharks are usually, you have to get like fetal specimens. Um, or there's like very few sharks you can get like chain cat sharks are like this big, like 2 feet long as adults, maybe 3 feet long. If I get a baby one of those, I could say it and it's saying well, but a baby, a 5 foot long white shark as a baby is going to be too big to stain, and so I'm limited in the diversity. But I've yeah, I'm limiting the diversity, but I do want to try staining more cartilaginous fish. These skates that I just happen to have, we order them from babies as babies from an online store, a biological supply company that we got for our research, and at the end of their research, some of the ones died, and so we preserved them to add to the collection, and that's how I got those specimens.
well, we had some baby dogfish in utero from the from a supplier,
Mm-hmm.
Carolina Biological Supply too. So
Mm.
anyway.
I've got one or two of those around. A lot of times they're not in as good condition as I'd like them, uh, and so or they're a little too chunky and thick to work with. So I like to avoid using them when I can. That's sorry, not avoid it. I haven't found good quality specimens of those fetal dogfish, and the ones I had in my personal collection that I collected a long time ago are not good enough quality. They've kind of broken down given they were collected in a field lab um, offshore on a tiny rock, and we were dissecting these, and we had them in utero. And then all we had to preserve them was like a bottle of vodka, and we poured them in there, and that didn't preserve them as well as I could have. But it's what we had to preserve at the time. You got to
Right,
improvise.
we got to use, yeah. So Ned put there's tiny needlefish were seen 60 years ago off the piers at Pope's Creek on the Potomac. So you might want to go
Mhm.
check that out.
Yeah, no, I believe it. They needlefish can tolerate a pretty decent uh amount of fresh water and go and they can move, especially in the summer, they'll come up higher. And in the summer when there is less runoff from less and higher salinity events, they'll move further up rivers. Like I've seen them in ponds that are connected by like culverts in Florida, in fully freshwater ponds that they can tolerate pretty well. But yeah, they're around here.
Alan, you have your hand up.
Yes, I'm obligated by an unwritten contract to ask this question at the end of every session. You did sort of explain how you got into this immediate work that you were doing sort of during college. I'm interested to know sort of just how you got into the field, at what age, were you very young, or sort of where you got into this area that interested you. And thanks so much. Just
Mhm.
fascinating and beautiful.
So to answer that, I'm going to read. I was debating putting this in here, and I ended up not. But I'm going to read I believe it's my third grade paper, and the third grade assignment that I did, and the comments that the teacher gave with it as my favorite vacation is Martha's Vineyard. I went all over town. My favorite part was when I went fishing. The fish I caught were a bonito, bluefish, thresher shark, scup, flounder, and blackfish. I went swimming a lot in a lot of pools and in the ocean. I went to a lot of stores and restaurants. Every day I went fishing and to the beach. The favorite thing I did was when I caught a thresher shark. And the teacher wrote concluding sentence question mark. And she's wrong. I caught a thresher shark is a concluding sentence. But she also wrote, no, I get the feeling that you like fish and fishing. I'm very happy that you have a hobby and interest. However, you need to write about different topics. No more fish stories. Uh, let's talk about neater handwriting to see me. Anyway, so I clearly did not follow her advice, and I continued writing about fish for a living. And so honestly, in hindsight, she was probably a pretty bad teacher. She's telling students stop pursuing your interests because this was also the second week of classes based on the date. So I must have talked about fish so much in just 2 weeks that she was already fed up with it. And then she's like, no, no more fish. And so anyway, if you're a teacher out there, encourage your students to pursue their interests and not shoo them away from it. Because I write about fish for a living. But I was hooked on an inexplicable hyperfixation on fish from an early age, probably because I'm neurodivergent and there was no rhyme or reason. I was just born and fish, fish, fish. So there's no explanation for it. The way I got into, I thought I was going to do something like Shark Week stuff based on that was the only thing on TV that I saw about fish. And then, long story short, I found a fish in the hallway freshman year at a field lab and asked how it got there. And that was the story I told it when I back in my amphibious fish presentation, and that kicked off my field in amphibious fishes, and I got that tattooed on my arm as well. Yeah. So I don't know how I got in this field. I was born and then I was in that field.
Wow. What great answer.
And we're so glad that you did, and that you didn't follow the teacher's directions with that. So you wouldn't be here doing what you're doing and sharing what you're sharing with us tonight, and helping us understand fish and their place in the ecology and natural history, which is wonderful. And how did you catch the thresher shark?
Oh yeah, so that's still one of my favorite catches, and one of the rarest catches probably I got. I mean, people get bigger thresher sharks like usually like offshore, like you get them fishing, like people are common. People get them Ocean City. I got like it was like a 2 foot thresher shark. It was like very small thresher shark that must have been like a newborn in the summer in Martha's Vineyard. That's like they move up north and give birth in the summer, and then there's migratory patterns. And it must have just been a baby that was recently born. And even back then, because of my hyperfixation on fish, my hobby, like I remember in second grade, the year before that, I gave a book report on Audubon's first field guide to fish, where I just listed fish, descriptions of them, and why I like them while people were talking about Captain Underpants. So I was familiar with, and I'm like, this tail is just as long as the body of the fish, and the face is all weird and scrunched up. This must be a thresher shark. And so I knew that as a young kid, and I have not caught a thresher shark like that. Whenever I tell any other person, fish biologist or scientist, this story, they're like, I caught a thresher shark is a concluding statement. That is awesome. And they give me a high five. And so that's how that works out.
Well, I agree. I think it is a concluding statement. And thank you so much, Noah, for sharing your art and science with us. We're all about the intersection of art and science. Science has been inspired by art and continues to do its thing, and art is inspired by science. So we love what you're doing, and you have the websites out there. And I know that you and I will be in discussions on how to bring more of the clear and stain stuff to our museum over here as well. So we're excited about it. And everybody, stay well, stay curious. Goodness gracious, we got a big storm coming our way, so stay safe, everybody, over the weekend. And we'll get back together, not next Thursday, but the Thursday after that, and we'll go head into space. So come on back for Must Learn Thursday. We go all over and keep our curiosity muscle in shape. So take care, everyone. Good night.
Good night, everyone. Pleasure chatting with y'all.